introduction. I'm always happy to be back in our webinar series, and we've had a lot of business-focused webinar events recently. Today, we get to return to things near and dear to my heart. We get to go back to the lab bench and to the processing environment for a presentation by Bruker, specifically from Dean Roberts, who is Director of Technology and Market Development at Bruker Applied Spectroscopy. Dean focuses on expanding the use of FT-NIR, FT-NIR, and Raman spectroscopy for quality control, process monitoring, and other industrial applications. Dean is a chemist. He obtained his BS in chemistry from the University of Wisconsin-Madison in 1982. On his career path, Dean cut his teeth in quality control laboratories before joining Matson Instruments as an application scientist working with FT-IR spectrometers. He joined Bruker Optics in 1999 as the Midwest Regional Sales Manager and has been moving up the Bruker ladder ever since. Dean's current areas of interest include development of new applications for both benchtop analyzers and inline systems, hence the topic of today's webinar, for the dairy and biopharma manufacturing operations. Dean, we've had you on before and happy to welcome you back today.
Thank you, Andy. If you'd like to go to the next slide. Okay, I noticed that I left something critical out of the title here, and that would be near-infrared spectroscopy. So that's going to be the topic today. We're also working with Raman spectroscopy for process analysis with certain products and unit operations within the dairy industry, but the near-infrared is the one that we've been working with the longest. Andy, if you'd like to progress this. So looking at the agenda, I want to look at first what's unique about the dairy market compared to some of the other markets that we do process analytics in. Bruker is active in pharma, chemical, petrochemical, petroleum refining, pretty much anything where a product is made and marketed. At some point in the value chain, there's a near-infrared spectrometer, whether it's on the benchtop or whether it's in an inline process analysis. But the dairy market is unique, and we'll talk a little bit about what makes it unique. And there are today changing economic factors that are driving the interest in PAT for the dairy industry. Some of the larger plants have been looking at that as a way to get more consistency into the product that they're making. But the economic factors that we're looking at today, particularly in the cost of protein, are really driving the industry towards these inline analyzers. So then we'll go on and we'll look a little bit at the performance of FT-NIR analyzers relative to the reference method. I know many of the people that are on this webinar today are used to using FT-IR spectrometers for compositional analysis of milk. And I want to talk a little bit about what FT-NIR brings to the table in terms of those analyses and what the benefits are relative to establishing an inline method with a near-infrared. So then comes the big question. What possessed us to think that we could use calibrations from a benchtop instrument on an inline system? And I will tell you right now, this is a work in progress. But when I started talking to some of my colleagues, they said some things that at times were a little bit unkind, like, are you delusional? And they were all along that area. But then when I showed them the data, they kind of paused a little bit and said, maybe they got something here. So we'll look at the design of the flow cell that meets the 3A specifications for use in the dairy industry. And the design of that flow cell is critical to being able to use the benchtop calibrations on the inline system. Then we'll follow that up with the test that we did in a pilot plant measurement with Daisy Brands down in Garland, Texas. We'll look at the second test that was done in milk receiving, and then we'll look at future tests in the summary. Next slide, please. There you go. So the dairy market, it really is a unicorn compared to all of the other that we work in. Milk pricing, as you all know, is largely based on fat and protein, and the margins are thin. And if you look at the products, the USDA, for many of them, for processed dairy products, has established standards of identity. Now, unlike other markets, the reference methods for compositional analysis of milk products are exceptionally good. If I were to go out and tell somebody that is measuring feed ingredients for animal feeds that they could do protein at 0.2 or 0.03% accuracy, they would look at me like I was from another planet and didn't really know what I'm talking about, because in those it's anywhere from 0.5 to 0.7%. And if you were running your dairy that way, you'd be out of business in no time at all. But also, milk is a natural product. It changes with a variety of factors. So the forage base, the lactation stage, the breed of cattle, all of those things have an impact on the composition of the milk, which impacts the calibrations. Next slide, Andy. So talking about the standards of identity, this is taken from the USDA Standard of Identity for cheddar cheese. And you notice in red there, it says contains not more than 39% moisture, and in the water-free substance contains not less than 50% of milk fat. Now, that 50% of milk fat is the minimum, but you also don't necessarily want to be over-formulating your cheese so that the butter fat goes much higher, because you're giving away a fairly high-cost component. And many of the smaller cheese plants don't actually standardize their milk before it goes into the vat milk. And there have been several presentations in industry where they've talked about the changing fat to protein ratio in milk as we get cows that produce more and more butter fat. And the increase in the protein production of the milk does not really follow the increase in the fat. So there are opportunities there to be able to standardize the vat milk to make sure that you meet the standard of identity, but not over-formulating. Next slide, please, Andy. So the milk protein. You saw there briefly the numbers of proteins, and anybody that's walked through Costco or a health food store knows a variety of protein sources that you can buy, and much of it comes from the whey protein. But looking at the April prices from USDA, milk is at, for the class 2 milk, is at $18.82 per hundredweight. But looking at the butterfat price and the protein price, you see that protein is actually eclipsing the butterfat in terms of the price per pound. So the increasing value of that protein, that's in many instances is coming straight from the whey processing, is really driving the interest in inline formulation. In fact, there was one person that told me that if they could save 0.5% in the protein value for the samples that they're formulating, they could pay for a $200,000 process analyzer in as little as 2 months. The ROI on these things is huge. Next slide. So this actually comes from a presentation that was done at Cheese Expo a few weeks ago by Eric Miller from High Ground Dairy. And here you can see the price trends over the last few years for the different whey samples. You've got the dry whey, the whey protein concentrate, 34%. But on the right-hand side, you see the WPC 80 and the WPI 90. And if you look at those prices, how they're skyrocketing over the last few years, the expectation is that this will actually tend to increase, which makes being able to do inline analysis of the product that you're making even more and more attractive to companies to justify the purchase of what really are pretty expensive analyzers. So as we see these prices going up, we see it going into the production of the main product, be it cheese, milk powder, any of the yogurts or sour creams, anything that's a cultured product that is producing whey is going to be a candidate for looking at these inline solutions. Now, in terms of the Bruker product line, some of you may be familiar with it, others may not be, but basically this product series is based on FT-NIR spectroscopy. And in looking at the vibrational spectroscopy, which is what an FT-IR is a member of, we also have Raman and we have near-infrared. So the signals that we're measuring with the near-infrared spectrometer are basically overtones and combinations of the signals that are measured by an FT-IR. There are some benefits to an FT-IR, one of which is the ability to use a flow cell for the measurement that has a significantly larger path length than the FT-IRs use. And the flow cells are made from quartz, which does not wear over time, which eliminates drift. So moving from the left to the right, we've got the MOVE-T dairy analyzer, which is dedicated for liquids. We've got the MPA3 dairy analyzer, which is a hybrid system that can measure both solids, cultured products, powders, and the fluid dairy products. The Tango R is a benchtop instrument that operates in reflection, and that is used to measure powders or cultured products or cheese products and other solids. The Beam is one of the inline analyzers that uses reflection spectroscopy, so that can be used for measuring high solids products like cream or cultured products and powders and cheeses in an inline analysis. And this is a single point analyzer. And then finally, the Matrix F2 is a 6-channel multiplexed near-infrared analyzer that's capable of measuring both liquids and solids. Now, in terms of certification of near-infrared for dairy products, you've got 3 documents that are shown down on the bottom that are IDF and ISO that talk about the approval of using near-infrared spectroscopy for both the condensed phase and liquid products in the dairy. Next slide, please. All right, so looking at the repeatability and the accuracy of the near-infrared, I wanted to throw up the accuracy here for the Majanier method for doing milk fat. And these are developed by round-robin studies, taking a number of laboratories, throwing out the low, throwing out the high, and then looking at the average values from each one of the laboratories. These are the methods that exist for fat, for protein, for total solids, for lactose, and these are the basis for all calibrations of near-infrared spectrometers, FT-NIR spectrometers, Raman, or any other technologies that are used for measuring dairy products. And the critical values here are the S sub small r, or the standard deviation of the repeatability, which means you have one operator, one instrument, one set of samples, run multiple times, and what is the standard deviation? And then S sub large r is the reproducibility. So it's the same thing, but it's using different instruments and different operators on the samples. And those are really the limits to what we can expect for the reference values that are used to calibrate our spectrometers. Next slide, Andy. So this is an example of our MPA spectrometer. And on the right-hand side, you see the true value is measured by DQCI using the Majanier method. And you can see the predicted value there. And then in the center, you see at the top, the fat with an RMSEP of 0.02%. And then the protein RMSEP is also at 0.02%. So the accuracy of these calibrations is right in line with the repeatability and reproducibility of the standard method that is being used for that, which tells us that the accuracy is about as good as it can really get in terms of comparison to the reference methods. Next slide, Andy. Thank you. But accuracy and repeatability obviously are not the same thing. So this is an experiment that we did on one of the MPAs where we took some samples, put it in a 600 ml beaker, and we started measuring replicate measurements. So each one of these dots is a 5 milliliter sample volume pumped between the measurement cycles on the instrument. And you can look at it, and we've got about 0.007 repeatability right up to where we added the fresh milk to the beaker. Now, this beaker was sitting on the apron of the instrument. It was cooling off a little bit because it was at 40 C when we first put it on the instrument. And as it was cooling off, then we dumped some more milk into it, which cooled it off a little bit further. And temperature does have an impact on those measurements. And you see that that drop of 0.005% is easily seen. So the repeatability of these benchtop near-infrared spectrometers is really, really quite good. And that is one of the things that allows us to think that we can transfer a benchtop calibration into an inline system. So in each of the measurements, we co-added 256 scans, which takes about 30 seconds, actually 43 seconds. And we just kept pumping and scanning for the duration of this experiment. Next slide, please. So I was at a conference called IFPAC, and IFPAC stands for International Federation of Process Analytical Chemistry. It's largely made up of pharmaceutical scientists with a few scientists from the chemical industry thrown in, and a pretty healthy contingent of scientists from the FDA. And the whole purpose of this conference is for the different scientists to get together and share notes about what they're working on, what their challenges are, and trying to come up with a better way to do the measurements that are made on the products that we all use on a daily basis. It's a very good conference. In fact, the pharmaceutical companies are allowing their scientists to come out and talk specifically about things that they're doing in their quality areas to qualify products, what they're doing for in-process testing to ensure process consistency and product consistency along the entire value chain. And FDA is there to say, yeah, we like what you're doing, or yeah, maybe we want to have a little bit of a change. So I ran into another company that was exhibiting there. It's Custom Sensors and Technology from Fenton, Missouri. And they had this little flow cell on their booth, and it kind of caught my attention because I'm used to the big honking things that we use in process and analysis with probes and flow cells and other different types of sampling interfaces. And they started talking about the repeatability of this. And the nice thing about that is this little flow cell showing there in white is actually made out of polypropylene. And it's considered a single-use device where it's about $200 to put that in. And I bought one. And we put it in a peristaltic pump, closed cycle loop, and started running milk through it and making measurements. And I used the benchtop calibrations simply because I didn't have any calibrations that were developed on this flow cell. Next slide, Andy. So we started off with skim milk, 1%, 2% whole milk, and then we got, sorry, my Dexcom is going off here. All right, I silenced it. We started off with the pasteurized homogenized milk, and then I got some milk from one of our customers locally here that has a higher butterfat content. And then I spiked that with cream to really pump it up there. Now, in some of the existing technologies we've been using for analysis of raw milk, after you get to about 3.5% butterfat concentration in the milk, you're pretty much done because the fat droplets scatter the beam so severely that you just run out of signal. And I wanted to see if I could take this flow cell to a higher level of butterfat content. And here you can see the measured value, the true value, and the difference using the benchtop calibrations. Now, one thing I would point out is that the raw milk and the spiked raw milk were not homogenized. And the degree of homogenization makes a big difference on the predictions for the fat and also for the protein, but not as much for the protein. And because fat and protein are key components of the solids measurement, it also impacts the solids measurement. And what we typically see on our analyzers is that if the fat droplet size becomes larger, the prediction value for the butterfat will be higher. And this is actually reflected quite nicely in the measurements that we did. So looking at the differences, I was at first in a little bit of disbelief. I didn't think that this was real. There's got to be some kind of an accident there. So I did it again, and I got the same thing. Now, these samples were all measured at 40 degrees C. I stuck a gallon bucket in a water bath at 40 C. I let it temperature stabilize, measured the sample, and did that for each one of these different samples, a skim, 1%, 2%. And there is actually no difference between the measured and true on these things. Now, I would expect a little bit more. In fact, on the 2%, you see a little bit of a difference on the difference, which is 0.1. And I think that was probably a temperature outlier, but I don't know for sure. On the second run, I actually didn't see that. Okay, Andy, next slide, please. So we thought about making a cell for the dairy industry. Now, this one for the peristaltic pump is really used for more of a benchtop type application or small pilot processes because it's only got a 1/4 inch input. And we know that the piping that's used in a dairy plant can go anywhere from 2 inches and larger. So we wanted to get a flow cell that was 3A certified for use in the United States. And we wanted it to be scalable to match existing pipe sizes. So if you take a flow cell that's smaller and you have to neck it down, it really messes with the flow measurements that you're doing on these systems. And if you neck it up, it has the same impact, but the other way around, where it goes to a lower flow measurement. And one of the things that we found with the fiber optic probes that we've been using to date is they use internal fibers to move the light through the sampling gap and then back up to the instrument. And when you have two parallel surfaces that are separated by something that has a different refractive index, in this case air, you get what's called interference fringing. So it's a cosine wave pattern that sits on top of the spectrum. And because of the high optical density of a milk sample in the probe, those interference fringes constitute a fairly large portion of the signal. And if you take the fibers off and put them on, it changes the period. And that makes it nearly impossible to transfer a calibration, not only from instrument to instrument, but from probe to probe. And the amount of money that goes into putting together an inline calibration is significant, to say the least, because you have all the reference chemistry that you've got to do, and you just have to collect so many samples to be able to get the range of values necessary to have a calibration that it becomes cost prohibitive to do so. We have to be able to maintain the accuracy and the repeatability at different flow rates. And that would mean, does the cell contribute to turbulent flow? So when we put a fiber optic probe into a 2-inch pipe and that probe has a 1-inch diameter, it is going to mess with the flow pattern and it's going to generate turbulent flow, which will have an impact on the measurement repeatability. We have to be compatible with caustic CIP cleaning and in some cases SIP. It's got to be easy to install and easy to remove. And then based on the previous work, we added the following criteria in there. We have to be able to use the calibrations from our Benchtop FT-NIR for the inline measurements, and we need to be able to use the Benchtop near-infrared spectrometer to serve as a validation reference for the inline measurements. Next slide, please. So why do we want to use the Benchtop for the inline measurements? Number one is developing the calibrations. We already have mature calibrations for the variety of the dairy products that we're measuring on our Benchtop instrument. So we're doing cream, we're doing milk of all the different varieties. We're looking at whey products, we're looking at ice cream premix, we're looking at the dressings for cottage cheese, we're looking at the base for doing sour cream. All of those things are liquid. Because of the larger path length, we can pump them through the instrument without any problems, and the calibrations are quite nice. But we also then have the ability to spike samples or be able to change the relative concentrations of the constituents in the sample. And that is done to break up collinearities and make a more robust calibration. Another big criteria is that there's no sampling area. So sample area is when you are taking a sample out of the process, doing the reference chemistry on that sample, and then trying to equate those results back to the spectrum that was measured by the analyzer. The sampling error is the time difference between when you actually pulled that sample and the spectrum that you measured. And if you have a very consistent process, then that's usually not a very big deal because this process doesn't change too much, but it can happen. And if you have sampling error where it just doesn't match the calibration, you have to be able to go back and justify why that sample is different in terms of its prediction. So you can take it out of the calibration and be able to continue developing it. And quite frankly, anybody that has developed a calibration by taking samples out of the process and using the spectrum developed by the process analyzer to develop the calibration knows how tedious a process that is. And quite frankly, it just makes the commissioning time for an inline analyzer so long as to be cost prohibitive. And that in itself is one of the reasons why people don't adopt inline analyzers. It also significantly speeds up the installation and commissioning of the system. So if we can start with a calibration that's going to work on all of the flow cells with all of the instruments for the same unit operation, then we can go in, we can put it in, and instead of calibrating the instrument, we're validating it. And we can get that done in a fairly short time. And by using the Benchtop instrument as a validation source, we can get those results much, much more quickly than using the reference method. So we do one measurement that takes 30 seconds rather than 5 measurements for the protein, the fat, the total solids, the lactose, and we can get it done much faster. Next slide, please. So this is the 3A flow cell that Custom Sensors and Technology came up with. And it's certified by 3A for diameters from 1/4 inch, which would be just very small lines and maybe a pilot type facility, all the way up to 6 inches in diameter. It's currently made from 316L, but we're also looking at making a cell out of Pastille C276, because in some of the European factories, they're required to use hypochlorite as their CIP solution for raw milk. And 316L will not stand up to hypochlorite as a cleaning solution. We've got triclover sanitary flange fittings, and there are no fiber-to-fiber connections inside the flow cell. Next slide, please. Here are the drawings of the flow cell, and you can see that the topmost on the left is showing you a down the throat view. We have a 1 millimeter path length that is adjustable to 1 millimeter plus or minus 0.04 millimeters. And we actually use a measurement of ethyl acetate as a solvent and compare the peak intensity to verify that the path length is in fact right at 1 millimeter. We've got standards that we've set up for it, and then we compare the absorbance spectrum of each one of the flow cells to the standard, and we can adjust the path length in the factory to minimize those differences. That is one of the things that helps transfer calibrations from one instrument to the other. It's very symmetric, and we've got 3/4 inch NPT fittings on the SMA connectors for the fiber optics, which allow you to put a flexible conduit fitting on to protect the fiber from washdown and any sanitary processes going on in the plant. That flexible conduit would then go into a junction box where it transitions to a rigid conduit to protect the fiber. Next slide. So the first test that we did was at Daisy Brand in Garland, Texas. And they've got a beautiful pilot skid there. I want to thank Fernando Munoz for hosting us to do these tests. So in the leftmost picture, you see the matrix F, the fibers, and the computer sitting on a cart. And that black spool at the top is the spool where the fiber is coiled up. And we used 50 meters of fiber to go over to the flow cell and 50 meters to come back. And fiber length actually has an impact on the long wavelength cutoff of the measurement and on the signal throughput. So we wanted to use something that was going to be a length of fiber that was reasonable for installation of a multiplex system in a dairy product. And on the right-hand side, you see the flow cell in the red square with the fibers connected to it. So in this case, we actually had to neck up the pilot facility and the tubing to fit the 2-inch diameter flow cell that we were using. And the milk was sitting in the bulk holding tank, and you see a little bit of foaming on the top of that. So this was the skim milk, and we'll talk about that foaming in the next slide. Next one. All right, so this is the experimental design, and this looks complex, and I will tell you that it's far more complex than what this even looks like. I just didn't have room on the slides to go through all of it. But what we basically did is we added skim milk to the bulk tank, and we recirculated it at 10 gallons a minute with the heat exchanger in the pilot skid to set the temperature to 5 degrees C. Then we turned the recirculation pump off. We did a static measurement, and then we turned it on and set it at different flow rates. So it was 30, 50, and 80 gallons per minute. And at each one of the flow rates, when it stabilized, we measured the near-infrared spectrum. And then we turned the flow rate down to 10 gallons per minute, changed the temperature of the heat exchanger to 40 C, and we repeated steps 3 through 7. So we did a static, a 30, 50, and 80 gallon per minute measurement. And then we did the same thing again, going back to 5 degrees C, but this time we added butterfat to get to a nominal 1% fat level. Went through the whole thing with the static measurement, 30, 50, 80, turned the temperature up to 40 C, 30, 50, 80, and then we repeated that for 2%, 3%, 4%, and 5% butterfat. So I was going to go back and try and figure out how many measurements we made, and that was just too confusing for me. So we made a bunch of measurements. Next slide, Andy. So we looked at the measurements, and these are the results from 5 degrees C. And you see immediately that there's a slope and offset there. But also on the skim milk and then the 1%, you see that those numbers are really a little bit off from the predicted versus actual line. And the reason for that, we believe, is foaming of the whey proteins. You saw the foaming on the top of the bulk tank, and it did have a bit of an impact on the flow rate. So down below, you see the 5 degrees C measurements for the static, 30, 50, and 80, and you see the average. And on the skim milk, we were a little bit below what the actual measurement was, and we were a little bit below what the actual measurement was for the 1%. But when we got up into the 2% and higher, it compared pretty favorably to the actual measurement. The 2% was a little bit high, and then we continued to be a little bit high. And what we saw was there was a slope and offset there. And our calibrations for the benchtop instrument are all measured at 40 C. So our conclusion there was maybe the slope and offset is due to the temperature difference between the 5 degrees C and the 40 degrees C calibration spectra. And what we do know is that temperature has an impact on the centroid of the water vapor peak or the water peak, and also on the symmetry. So we know that temperature does impact the predictions. Next slide. So here we looked at the warm milk measured at 40 degrees C. And we still see the impact of what we think is the foaming at the skim and at the 1%, but the other ones line up perfectly with the predicted versus actual with no slope and offset correction. And when we look at the different flow rates, we see that the 80 gallon per minute is quite a bit off from the actual value, and we're reading a little bit lower on those ones. But when we get up into the 2% through the 5% levels, we're really, really close to the reference value at the different flow rates, and it looked really good. So our conclusion here is that it's not the flow cell that is actually causing the changes in those values, because if it were the flow cell and it was turbulent flow being developed by the flow cell, we would have seen that in both the 40C and the 5C. So we're thinking that that slope and offset really is due to the temperature. Next slide, Andy. So here's kind of an example of what we got with the 40 degrees C. And there was a little bit of a difference in the slope and offset. We didn't put the skim milk in there, and we did put the 1% in there. But then we show the results that we're getting with the slope and offset. And the 1% was still a little bit high, and the 2% was a little low, but we're right on there with the the 3% through the 5% measurements. And we had enough signal left there where we think that we probably could have gone up to 6% butterfat quite nicely with this. Next slide. So here's what we got at the 5 degrees C, and then applying the slope and offset. You see really kind of the same relationship that we had with the 40 degrees C after the slope and offset correction is applied. Could we do a little bit better on that? Maybe, but really it's only the 2% sample that's off, and there may be some residual impact from the foaming there that hasn't been completely emulsified by the higher fat levels. Next slide. So looking at the total solids, we know that the protein and the fat values have an impact on the total solids because those really add up to, I think, the largest percentage other than lactose. And we see that the total solids kind of follows the same trend that we had with the 40 degrees C for the skim and the 1%, and then it starts to come back into the linear relationship at the 2% level. Next slide. And then here's total solids with the cold milk, and here you see very clearly the slope and offset difference caused by the temperature offset. So if we look at the difference versus the predicted, it's a linear relationship, and linear relationships are nice because they can be pretty easily corrected. And on the bottom, we see the slope and offset corrected. Next slide. Thank you. So the next step was to go into a real processing plant. And again, we came in here with the 2-inch flow cell. And the reason we used the 2-inch flow cell for these 2 trials was it's the only one we had. We hadn't yet taken delivery on the 3-inch, and we're still waiting on delivery of our first 4-inch cell. But the Matrix F2 is the process instrument. It was put into the control room where we had stabilized temperature for the spectrometer. We had 50 meters of fiber out, 50 meters back, and we put it in a neck down section of a 4-inch pipe. The sample temperature is not controlled because this is the milk tanker unloading. Next slide. So here are the results that we got for a number of samples that were measured over the course of unloading one tanker. And you can look at the raw near-infrared value and then the slope and offset adjusted near-infrared value and look at the difference. And it's staying right in that 0.03 to 0.04% with a couple of exceptions throughout the course of unloading that tanker. And what you do see is that the fat level increases as you're going through the unloading process, indicating some segmentation in the tanker truck itself, which is one benefit of doing inline measurements. And there was actually a paper published by Hilmar a number of years ago where they pointed this out quite clearly, that there is segmentation in the reference values for the measured values for fat as that tanker is unloaded. And if you'd like to get a reference to that publication, send me an email and I'd be happy to send it to you. Next slide. And then here's the protein. Now this actually looks pretty reasonable. And just as a point of context, the reason that we didn't do protein in the first study on the pilot skid is the results just didn't really change enough for us to make any decisions about whether it was working or not. It's kind of like doing a single point bias adjustment. It's right at one point, but you don't know if it's right at the rest of the points. But here we were measuring the protein, and you can see that it's got a pretty reasonable repeatability and comparison to the benchtop values that we used as the reference lab for these measurements. Next slide, please. So what this looks like to us, and at least for fat and total solids, using those benchtop calibration is going to work, provided we know the temperature and that we can apply a different slope and offset correction to account for the temperature difference between the benchtop calibrations at 4DC and what we're actually measuring these samples at. If we're in a process where you're using a heat exchanger, it's a pretty good given that the temperature is not going to have much of an impact. We can't really say for sure that we're going to get the same temperature when we're looking at milk receiving, because each one of those tanker trucks is going to come in at a different temperature. So we may have to put a temperature sensor in line and then dynamically apply a slope and offset to the predictions that come in to try and normalize that for the temperature differences. Another option is to move to a PLS2 calibration where we can use temperature as a variable. So these are some of the other things that we have to use to try and really nail down the ability of this to predict fat. We also need to repeat the pilot skid application where we're varying both the fat and the protein. So what we'd be doing is adding UF skim to that milk to change the protein content, and then adding cream to that sample to change the fat content. And we would do so that the fat and the protein do not change in a collinear manner, which would mean taking the number of steps that we have in that analysis that was in one of the previous slides to a much higher level and doing a lot more measurements to be able to validate that we can do the protein that way. And for standardization of milk in milk processing facilities that are making a cultured product or an acid set product, we're going to have to make sure that we can get the protein right, because the protein to fat ratio is going to be critical for doing this. And then as we move into the whey processing for the high value WPC 80 and WPI 90, getting the protein right is going to be absolutely critical for doing the predictions of the protein to try to reduce the amount of giveaway in the sample. We are also looking at any process testing that any of our customers can come up with that they think might be a win for them. And please consider this an open invitation to share your ideas with us, because we're up for trying just about anything. And as we build this market, we're going to be looking for more and more applications to be able to apply this technology in the dairy industry. Next slide, Andy. So to try and wrap this up, is it feasible? Is it practical to extend a benchtop calibration to the inline case? And in what we've tried so far, yeah, we think it is. And we're going to be doing some installations with customers who actually agreed with us and have purchased systems to go inline. And we will be working with them very, very closely to commission these process analyzers, make sure that they're giving them the correct results, that they're giving them the right repeatability, so that they can have actionable data to control their process and enhance the consistency. We're pretty sure that the temperature changes result in the slope and offset. I think we've proved that, and we're going to work on applying those temperature changes. Protein is still a work in process, and we think it's going to work based on some of the results we got at the milk unloading, but we really need to get a wider range of protein to be sure that that's what's happening. And then we need to extend that work into the whey processing, where we're looking at higher and higher levels of protein. We need to be able to look at those more viscous samples to make sure that the flow cell is not fouling. And if it is fouling, what recovery is needed to keep those results in line with the repeatability and the accuracy expectations. And then we're going to start trying to apply this to other industries. In fact, I gave a version of this talk on Tuesday to a group of pharmaceutical scientists. And the discussion after an abbreviated version of this talk went on for 30 minutes on where they could apply this in the pharmaceutical industry. So we think we've got something here that might turn into a big deal. With that, I'd like to conclude the presentation. I'd like to thank you for your time and your attention, and I'd be happy to answer any questions that you might have.
I'm back, Dean, and thank you so much for the presentation and also for tolerating my somewhat ham-fisted advancement through the slides as I tried to anticipate where your breaks would be. So I very much appreciate your patience with that. We have a couple of questions that have come from the audience, and we have a few kind of high-level questions that are good tee-ups. So we're just going to launch right into those. With the tanker truck protein example, you alluded to this challenge of developing calibrations when you have very little variation to work with. Can you elaborate on that problem, especially in the context then of this possibility to develop calibrations benchtop and transfer them out to the process?
Absolutely. If you're developing a calibration solely based on pulling samples from the process, doing the reference chemistry, and then using the reference chemistry with the spectrum that is measured by the analyzer at a similar time frame in the process, and you don't have a lot of variability in your process, you don't have a calibration. You can measure 1,000 samples, and if they're all the same thing, you don't have a calibration. If you have 100 samples and you have a reasonable range of values, then it's entirely possible to get a starter calibration. But to build that calibration out and make it robust, you need to be able to model the variation in the matrix that you're looking at. And we know that milk is highly variable. It's different protein concentrations, different fat concentrations. The fatty acid profile varies with the breed of cattle. It varies with the forage base that's being fed. So there are differences between the spring, summer, fall, and winter. And it also varies due to the lactation stage of the cattle. So if you don't model that source of variation, you're not going to be getting good predictions as the feedstock varies due to these reasons. In our fluid milk calibration, we've got about 6,000 samples in that calibration that represent all different types of milk, whether it is pasteurized homogenized milk, UHT milk, raw milk, differences in the lactation stage, the forage area, breed of cattle, area of origin, all of those things are represented in those calibrations. So if we can use the benchtop calibrations and the inherent variability of the samples that are present in there, it gives us a lot better feeling that the predictions that are coming off of these varied samples on the inline system are going to be accurate.
Yeah, so Dean, stepping back from those seasonality differences, for example, and some very dairy systemic influences on the validity of results, let's talk about temperature for just a moment. You referred to identifying that temperature was a factor, and you talked about the possibility of implementing temperature correction coupled to a temperature probe. Do you think that's a foregone conclusion? Is that going to be a new feature for this flow cell analysis going forward?
We have to figure out a way to do it, in all honesty, and I think it's going to be done through a Python script. But it is doable. And the other suggestion was using a method called PLS2, which is a method that allows you to put a variable in, put temperature in as a variable, and that becomes part of the prediction equation. So either one of those two results will work. If we're going to use PLS2, we'd have to write a Python script to do those types of predictions. But it is definitely a possibility.
Now, LinkedIn has been offering to train me in Python for a very long time. Maybe now is the moment to step up. Talk about the migration process. If you've got benchtop calibrations that are built, what's the user experience like then to translate that over to a newly installed flow cell?
We take the quant2 method, which is the PLS quant, and we put it on the process analyzer, and we tell the process analyzer to use that calibration. It's all a digital transfer. At this point, our target is to be able to use that calibration from the benchtop with no modification whatsoever. It may at some point be necessary to look at adding samples in from the process itself to try and tweak that PLS model in, but we are really hoping to try to avoid that if at all possible.
Got it. Thank you. I'm going to take one of the audience questions here. And I think the answer is implied by the fact that you were able to earn 3A certification and for a wide range of configurations here. The question was, does the flow cell get cleaned by CIP? Does it have any buildup inside that would contribute to specifically micro issues? In this case, was also part of the question.
Okay, so there are two answers to that question. One of which is the certification testing that the 3A inspector did. And what he did was fill it full of chocolate syrup and then have them run a simulated CIP through it, and then go in and swab to see if there was any chocolate syrup on any of the radii within the flow cell. We know that 3A mandates that there be a 0.25-inch radius on all junctions within the cell. And then he also took the swab around the silicon O-ring that seals the sapphire cylinders that constitute the path length. And he looked for any residual chocolate syrup in there, and he didn't find anything, which was critical to achieving 3A certification. We also loaned this flow cell to one of our customers who, because of confidentiality reasons I can't name, but they did swab tests on that cell after having been in the process for 2 days. And they were looking for any residual sample or any potential bacterial contamination. So they plated the swabs and put them in the incubator to try and grow them up. And they saw no evidence of any bacterial contamination or any residual product. The optics in the cell are sapphire, which they have to be in order to survive a caustic CIP, because if we were to use quartz optics, they would darken after about 24 hours of exposure to a caustic CIP.
Well, Dean, that's a perfect segue into this next question then, which is about the necessary maintenance for the flow cells. What does it take to keep these in good operating order?
Keep them clean. Don't break your fiber optics cables. Treat them well. And then every now and then, when you're in a plant shutdown, you remove the flow cell, clean it, dry it, and take a new background reference spectrum.
Something like an annual maintenance cycle. Is that a reasonable frequency for that sort of care?
I would do it whenever you shut down the process and you take things apart for cleaning and inspection and those sorts of things. You've already got the process down, so it's a good time to do it.
We had another very detailed question asking what communication protocols you're using to transfer the data between benchtop and inline spectrometers.
Well, to transfer the calibrations, there is no communication protocol because we're taking the digital PLS calibrations from the benchtop on a thumb drive and installing them on the PC that's controlling the process spectrometer. In terms of communication protocol of sending the results from the process analyzer back to a PLC or a DCS system or a data historian, we use pretty much any of the typical communication protocols. Our preference is to use OPC UA, which is an internet-based protocol, but we can use Modbus, Profibus. We can even go to 4 to 20 milliamp where it's hardwired. So whatever you happen to have in your plant is a protocol that we can use.
I think your answer had good refinements there. It's clear that when you have PC managed calibration data sets, that it's really a PC to PC kind of transfer. You can do it with a thumb drive, but in plant for communication protocols, it sounds like you have a broad range of options there. Let's move to applications. So you talked about looking at some other high value applications like the WPCs and WPI, but there's a lot of protein accounted for in permeate. Is permeate on your radar for application development?
All of it is. Absolutely all of it is.
I love that that was a short and to the point answer there. I'll ask you one final question, I think, barring further inquiries that come in from the audience. Is this ready for dairy? Do you have actual live systems working right now, or is there still development work to do before you're able to commercialize this while you're working with some trusted partners for that development?
We're going to be installing our first system in the next 30 days, and then there will be several systems that are going in right after that. With each of these customers, we've done the trial measurements, which is what was their justification for actually making a purchase. We will be working with them very, very closely. And the honest answer to that is we don't have anything that's been installed for a long period of time yet. As I mentioned earlier in the presentation, this is a work in progress, but I can assure everybody that this has our full attention and our commitment to being successful. Because we do really think that this is going to be a game changer for both Bruker and for the industry.
I see that Beth has popped back in because I said that was a final question. I want to build on that just a tiny little bit, though, Dean. You go back to 2019 and 2020, where the earliest IDF and ISO standards related to IR analysis were published. Do you think that this development to translate calibration from benchtop to process is the breakthrough that's needed to change the trajectory of adoption for the dairy industry? You think it is going to rapidly accelerate the use of this kind of technology in U.S. processing facilities?
Yes, I do. And our current thoughts at this time is that we should be using the benchtop near infrareds with the benchtop calibrations as our validation source for these inline instruments. You can't use an FT-NIR as a direct calibration source because the spectral range between the process instrument and the FT-NIR are different. And it just makes sense if you're using the benchtop near infrared calibrations to run the process analyzer to be using that as your validation lab as well.
Got it. Thank you so much. I think that concludes my role in today's webinar.